live. So we've been going through metabolism, and we were finishing up with the electron transport chain and recognizing that the electron transport chain does a couple of really critical things. It is responsible for reoxidizing, recycling all those electron acceptors. So we've got NADH is being reoxidized back to NAD. FADH2 is being reoxidized back to FAD. And a key point here, and this is kind of branching out of basic micro, but just sort of a general purpose point. We can talk about the idea of what we call aerobic respiration. That means that the final electron acceptor is O2. The chemical equation that we would kind of think about is we have the electron with oxygen. It is reducing oxygen, and we generate a molecule of water and the oxygen radical, or atomic oxygen. And so that is aerobic respiration. Now, for you and I, that's pretty much the only kind of respiration that really works. We don't have the ability to use things in the electron transport chain as the final electron acceptor. And this is one of the things that is really very cool and very weird about bacteria, is that not all bacteria are using oxygen as the final electron acceptor. If you recall, when we were talking about growth habit and things, we have a class of bacteria that are known as obligate anaerobes, in that oxygen is actually poisonous to them. They don't use O2 as the final electron acceptor. So they use maybe sulfur as the final electron acceptor. They may be they might use iron. There's a variety of different atoms and elements that you can use as the final electron acceptor that's not oxygen. So when we talk about obligate anaerobes, that's what we're talking about, is the fact that they're not using O2 as the final electron acceptor, they're using something else. In the eukaryotic world, that just didn't happen. I mean, there just is nobody you're going to look at that is a multicellular organism that is using anything other than oxygen as the final electron acceptor. So when we start talking about aerobic versus anaerobic respiration, that's what we're meaning, is that aerobic respiration is using oxygen as the final electron acceptor. Anaerobic respiration is using something else. All right, so just to refresh our last bit, if we look at the electron transport chain as it works, sort of diagramming what we just said, we have this business with these compartments, these two different kinds of spaces. We've got, in the mitochondria, this is actually happening within the mitochondria. Would be in eukaryotes. In prokaryotes and bacteria, we have this space between the plasma membrane here, which means that, by definition, everything in this side is cytoplasm. And we have then some kind of cell wall type stuff, either peptidoglycan or different kinds of carbohydrate wall, depending if it's gram-positive or gram-negative. And we have then this space that is referred to as the periplasmic space. It is just the region near the membrane, and so it's got its own little environment. And the electron transport chain is going to be situated such that we got complex 1, complex 2, complex 3, complex 4. And there are specific names for the complexes, but I don't think we need to worry about it. It is often referred to as complex 4. So different texts and things will talk about a fifth complex. This would be the ATP synthase. That is, i.e., the hydrogen ion channel that is coupled to the synthesis of ATP, or as I say, it is the ADP rephosphorylase, but whatever. This is referred to sometimes as complex 5. Just as a little point, you perhaps have heard of cyanide poisoning. Cyanide would probably knock back bacteria as well, more or less, although probably not as bad as it was eukaryote. Cyanide is sitting right here, and it is blocking electron flow. And this is a critical thought, is that we've got O2, or whatever your final electron acceptor is, floating around here. And the electron is actually skipping complex 2, but regardless, the electron is being dumped onto that molecule. And let's look to see what happens if we run out of oxygen. So we generate water and the oxygen radical. And for completeness sake, remember that each time an electron is passing in complex 1, 3, and 4, we have a hydrogen ion being pumped out. And so we end up changing the pH in these two different spots, in that we put a bunch of hydrogen ions out here and not very many here. And this generates a diffusion gradient that allows the hydrogen ions to flow back through with their diffusion gradient, and that's what drives the synthesis of ATP. So there are a couple of interesting biohacks you can do. One would be to put a block in this system. That'll screw things up. Another could be to put in punch holes into the membrane that would allow the hydrogen ions to slide back through. That'll screw things up pretty good. Because this is the primary ATP production system, or the ADP rephosphorylation system. We go from somewhere around 4 ATPs in glycolysis and Krebs cycle, and glucose should give you a total of 36. So that means that 32 of the ATPs are being generated in this specific spot. So this is the real serious energy workhorse system of respiration. So we have a couple of things then that we can begin to play with. One, what happens if you are an aerobe, an obligate aerobe, and the same thing actually if you're an obligate anaerobe. What happens if you don't have final electron acceptor? And this could happen because either you use up all the oxygen, or whatever your final electron acceptor is, or you're just in an environment that doesn't have much of that. If you run out, let's just start to see what happens. No O2 in the house. Well, with no O2 in the house, complex 4 can't dump its electron. Complex 3 is looking at complex 4 and says, here's my electron, take it. And complex 4 is going, I'm full up. I ain't got a place for it. Complex 3 is like, what the hell am I supposed to do with this? Complex 1 grabs an electron and says, all right, complex 3, take this. And he's like, I can't. Complex 1 is now sitting there with an electron going, uh-oh. Because here comes all these reduced electron acceptors saying, here, take my electron, take my electron. And it's like, ain't no place to put it. And very quickly, and with our cells, probably within 30 or 45 seconds, you suck up all of the free electron acceptors. They're all in the reduced state. At that point, your metabolism stops. Without free electron acceptors that are in the oxidized state, you can't proceed any further. This is a bad deal. We're going to have troubles now. So if you run out of electron acceptor, and this is the same for aerobes and anaerobes, if you run out of electron acceptor, you can't use this process. And this is like your number one ATP generating process. Life starts to suck pretty seriously. So we've got a slight twist, sort of a shunt, that we can do a little bit of work. And this is known as fermentation. And it's actually a terminology distinction that I think for a while I personally was not using correctly. Anaerobic respiration is using the electron transport chain but not using oxygen as the final electron acceptor. If we are running out of electron acceptors, we can move into a different spot that is known as fermentation. And fermentation is primarily, and I have to think about this a little bit in terms of it being a universal definition, but I'm down with it. We're going to use pyruvate as a final electron acceptor. Now you may or may not remember where pyruvate was. So pyruvate is the end result of glycolysis. Let's just go back through that real fast. We have the glucose molecule. We're going to add 2 phosphates from 2 ATPs. Split this into 2 sugars, or into 2 3-carbon molecules, 2 glycerin molecules. So we got the 3 carbon molecules. They are then being oxidized, meaning that we have to have an electron acceptor come in, pick up an electron. Same thing happening with that guy. Still got 3 carbons. The next step is the ATP regenerating step, where each one of these 3 carbons is able to catalyze and provide the energy for the phosphorylation, or the rephosphorylation of 2 ADPs to 2 ATP. And we end up with 2 3-carbon pyruvic acids, or pyruvate. All right. So now, if you don't have a good final electron acceptor, oxygen, sulfur, iron, something that you can use in the electron transport chain, you can always go over to here. And what you can do is now dump an electron on the pyruvate. We're going to actually reduce pyruvate. And in terms of systems, that's not a great way to operate, because you spent this time oxidizing to get to pyruvate. Now you're going to reduce it. You're kind of going backwards. But it does one specific thing for us. In order to reduce pyruvate, add an electron, we're going to take NADH and dump the electron there. It is now reoxidized back to NAD, and it can go right back here and just do a little quick circle turnaround. This is not doing a whole lot for the cell, but what it does do is maintain the 2 ATP net gain. Excuse me. So fermentation is not a very sustainable practice, because one glucose molecule, instead of being worth 36 ATP, is now 2 ATP. That sucks. B, the final electron acceptor, oxygen or whatever you might have, is set up in such a way that when it's reduced, it generates a molecule that's fairly benign. Like oxygen turns into water. Sulfur, I mean, I can't use it, but the bacteria that can, they generate hydrogen sulfide, a gas that just dissociates, it just dissipates. So the final electron acceptor is very specifically chosen, if you will, to generate a really benign reaction product. So what you reduce it to is a non-toxic waste. In this case, we're generating some fairly weird crap. You got 3 carbons, and so there's a variety of different things you can make. And depending on what kind of organism you are, you can make 1 CO2 and the 2 carbon ethanol. You could make a 3 carbon acid. You can make, and the classic is, of course, lactic acid. You can make all kinds of oddball organic molecules, depending on what sort of organism you are. And what you have done is, in fact, establish the basis for a whole gigantic range of technologies known as fermentation. So this is fermentation, where we're going to use pyruvate as the final electron acceptor. And it is a temporary fix for most organisms. You and I, if we go into fermentation, we begin producing lactic acid. And if you do this for too long, probably like, in real serious sense for maybe 10 or 15 minutes, you go into lactic acidosis. You throw your blood pH way out the window, and it is a real serious problem you're going to have. Additionally, if you do it for just brief moments, you build up lactic acid in your muscles, and this, of course, is what gives rise to muscle soreness. And you have to you can reburn it, but the lactic acid for us is a toxic metabolite. For organisms that are producing things like acids and alcohols and things, they can produce an amount, but after a while, it becomes toxic. So you may or may not know, like hand sanitizer, when people were talking about hand sanitizers, there are percentages of ethanol that are not particularly toxic, and in terms of like really denaturing stuff, you want to get above like 60%. Or maybe 70%, 141, 140 proof. But with wines, beers, yeast guys, they can't handle alcohol concentrations much above 20%. And so the environment that they're working in becomes essentially toxic. I mean, they basically are peeing in their own pool, and there's going to come a point where they go, okay, we can't do much of anything. Now, you and I, we die. Microorganisms pretty much will just shut down operation and just sort of like do nothing. And then when the system clears, oxygen turns back on, they'll start growing back up again. But what this means is that you can generate all kinds of weird secondary products. Bacteria can produce every kind of organic acid. Bacteria are known to produce things like nail polish remover. You can produce vinegar. You can produce weird things in soy sauce. And this is all because in their desperation to find a final electron acceptor of some kind, they start using all kinds of things. So that is, in a nutshell, metabolism. Now, there are a number of little interesting points that we will get back to PowerPointing on and see how we can fit them into this sort of model of what we've been talking about. Not to call out Tori, but having taken biochemistry, you can probably tell us that, biochemical pathways, there are a lot of them, and they can be really complicated, and that would be true. So there are all kinds of biochemical pathways floating around out here, and we're only touching on just a couple of them. Part of the reason this system is a complicated bee works is because we have the ability to branch off from these different reactions and generate pretty much every kind of biological molecule. Now, one of the things that's really cool about bacteria is that some bacteria are able to grow with a real simple carbon source, some of them even just CO2, but like the bacteria that we grow in the lab, they can use like glycerin or actually a whole range of very simple carbon sources, something that we probably can't eat. They can use just a simple nitrogen source, and they don't need to have what we would think of as a complete diet. We need to eat protein because we're limited in how much protein, how many different amino acids we can biosynthesize. Most bacteria can biosynthesize everything. If you give them a carbon source and you give them nitrogen, they can do it. They can make every single molecule they need. The pathways get to be pretty complex. They don't grow very fast, but they're going to do it. Whereas you and I, if you don't feed me the appropriate proteins, after a couple of weeks, I'm going to start to get pretty sick and have some weird conditions because I can't biosynthesize all 20 amino acids. I can only biosynthesize, 13 of them, I think. So, these pathways then allow for the production of every single biological molecule that a microorganism might need. Now, from another perspective, I often tease my son is that if he were to develop a food chart, pretty much the only food that he eats is fat and sugar. And so we have our, Krispy Kreme donut pixie stick diet, which is an interesting one to work on. We can talk about that later. But if we think about this from a bigger perspective, we're not only eating glucose, and this goes for bacteria and for you and I. We are breaking down a whole range of metabolites, everything from starches and sugars to things like fats and other molecules. And so we can fit in and digest anything that is digestible into these pathways. So sugars are going into glycolysis. Fats are consumed 2 carbons at a time off the fatty acid tail, and they're fed in to acetyl-CoA. And so if you can eat it, you're fitting it into one of these pathways. If it's sugar, you're starting at the very top. If it's a protein or a fat, you're starting at the Krebs cycle. So with proteins, if you recall the structure of amino acids, if you break them down into individual amino acids, the core of an amino acid is a 2-carbon molecule. You strip the R group off, you strip the nitrogen off, and you fit that 2 carbons right there, and you're off the races. So we can, under appropriate conditions, consume all these things. If you're going to be carrying out fermentation, don't try to eat fats or proteins because you can't carry out fermentation with those molecules. You can only carry out fermentation with something that can fit into glycolysis. All right. So this is just sort of a summary here. Glycolysis, we're making some ATP. The Krebs cycle is oxidizing the products of glycolysis. And then the electron transport chain. So we're going to, in aerobic respiration, use oxygen. And this is just sort of a review. Here we go. You do not need to know it nearly this detail. The main thing you want to know is what goes in and what comes out. What goes in is glucose and the ATPs and some NAD, and what comes out is pyruvic acid and 2 ATPs. All right. Again, the Krebs cycle, don't need to worry more other than what goes in and what comes out. Electron transport chain, it's hard to sort of do it as what goes in, what comes out, but what we can do is what is it doing? And that is reoxidizing electron carriers and using the proton gradient to generate ATP. Just as a little note, this guy, Peter Mitchell, that discovered this mechanism was for a long time thought of as a quack because he was proposing an ATP synthesis mechanism that had never really been seen before. That is, you can generate ATP by placing cells or mitochondria into a low pH solution. And yeah, that's what the cells are doing. But actually, you can take bacteria or your mitochondria and put them in a little bit of vinegar, and you can make ATP that way. This simply because the lower pH outside is generating this proton gradient that makes for the energy to drive ATP synthesis. All right. So as we say, anaerobic respiration. When we're comparing that, aerobic respiration, we're using oxygen as the final electron acceptor. Anaerobic respiration is now going to be using some kind of non-organic, non-carbon, we save organic molecules as the final electron acceptor. That is fermentation. Anaerobic respiration is using different kinds of elements, different kinds of ions. And one thing is, so you can use sulfate, you can use iron, you can use nitrate, nitrite. One thing is that why I'm partial to oxygen, and so are you, is oxygen being electronegative, it has a really strong pulling power, and it is able to draw more energy from the electrons passing across the different complexes because it is a gigantic sucking force. And nothing beats oxygen. Sulfur is not bad, but compared to oxygen, sulfur is nothing. And so if you had to choose an element to use as your final electron acceptor, oxygen rocks. Fluorine, I think, would be pretty good too, but fluorine has some nasty side effects in terms of its corrosiveness. I actually have to think about that. But yeah, there's these elements that are referred to as being electronegative, and I think fluorine is one of them. Anyway, so oxygen works really well, but other molecules will work. And this is what we refer to then as anaerobic respiration. We're essentially carrying out electron transport. The final electron acceptor is just something other than oxygen that's not organic. Fermentation is what we were talking about when we're going to use pyruvic acid. So if O2 is present, or whatever else you choose to use as your final electron acceptor, you can run through this entire system. If O2 or whatever it is you like isn't there, we can carry out fermentation. So the main point is you generate a little bit of ATP, you regenerate or reoxidize the NADH back to NAD, and you're able to continue glycolysis. So it's not great, but it's not dead. And you generate then a range of different products. And that's the nice thing about human biology. In fact, eukaryotes, you either are generating lactic acid, which is what all animals do, or you're generating carbon dioxide and ethanol, which is what plants do, and yeast and fungi. But that's it. With bacteria, of course, a bazillion different products. Everything, butanol, propanol, acetic acid, propionic acid, which smells a little bit like somebody threw up. Ammonia, lots of different things are generated by a range of different bacteria. So, just as an interesting point, we use fermentation a lot for foods. And fermentation also has some specific pathological effects. But when people are talking about the microbiome or your gut biota and stuff, a lot of what you're talking about is the bacteria that can carry out specific fermentation events. So we can provide or culture different bacteria to produce different kinds of molecules. So Propionibacterium are found in certain cheeses. Propionic acid is, well, it smells a little bit like throw up. I guess it also smells a little bit like Swiss cheese. Or Swiss cheese taken to its extreme smells like somebody threw up. Your choice. Lactic acid is being produced by a range of different bacteria. This is how we produce yogurt. This is how you produce soy sauce and a whole range of different things. Of course, yeast are producing carbon dioxide and ethanol. Some fairly nasty bacteria like Clostridium, which is one of the causative agents of botulism, they are some of the oddballs, and they can generate things like acetone and isopropyl alcohol. Acetobacter can generate acetic acid. And so when you're starting to make like pickles or sauerkraut is the one that's always thrown out, you make sauerkraut by taking cabbage, which has got a fair amount of sugars in it. You add salt to it, so you're increasing the salt concentration, and then you just let it go. You put a rock on top of it, so you block the oxygen, because you need it to go into fermentation. And the high salt is going to preclude some nasty bacteria, and the pH begins to drop because the bacteria that you culture are actually producing their own vinegar, which is kind of a cool little trick. So, one of the things when you have an anaerobic situation, there are a couple of really nasty bacteria that we'll be getting into, one of them being the bacteria that causes botulism. And so, improperly canned foods, and I would imagine improperly carried out fermentation, you could make something that would be really toxic. I suspect that it would smell horrible, and you'd look at that and go, I ain't eating that shit. But it is possible to poison yourself, which is not to say you shouldn't try fermentation. It's actually a lot of fun to ferment foods. That's what you get. So another way that we can generate energy is, of course, through photosynthesis. And we may or may not know photosynthesis, of course, in plants, in the chloroplast, but bacteria also carry out a photosynthesis of sorts. And sometimes the photosynthesis is actually similar. In plants, the photosynthesis is taking water and splitting it to produce an oxygen radical, and then the next water comes along and splits, and you get an O2 molecule, and you're pulling electrons out of the water. And these electrons are being used as the electron source, much in the same way that electrons are being used and pulled out from different foodstuffs. And you're generating oxygen. And so this is referred to as the oxygenic type of photosynthesis. Cyanobacteria are carrying this out. Anoxygenic is a kind of photosynthesis in which you're using hydrogen sulfide or other different molecules to split apart and extract electrons. And that doesn't generate oxygen. The cyanobacteria, when they came on the scene evolutionarily, they began changing the atmosphere of the Earth. And it's actually a really critical thing because when they first evolved, the atmosphere was primarily nitrogen, carbon dioxide. They start chewing on it for a few billion years, and they begin sucking the carbon dioxide out of the atmosphere and taking water from the oceans that they're growing in, splitting that and generating oxygen. So these are the photosynthetic guys, and they're kind of oddball fellows. Another set of really oddball organisms we've talked a little bit about, the chemolithotrophs. These are guys that rather than actually getting electrons from organic molecules, they're getting electrons from inorganic molecules, from minerals. And they're carrying out some odd reactions in which they can oxidize, remove electrons from minerals, and then shunt those electrons into electron transport chain. So you either have, the standard way we do it is electrons are coming in from these electron acceptors like NADH, or you can actually have electrons coming in from the minerals and the rocks and different compounds that you have to be sitting around in. It's not as effective. You don't make as much ATP, but it does work. It is a really weird way of thinking about extracting energy. So, all together then, we have, and it's one of the things that we start thinking about the way we work, is that we're so used to the lifestyles that we live, that is, you eat carbon, and that does two things for you, or carbon-containing foods, rather. Don't eat carbon. Eating charcoal doesn't work. You're getting carbon in, which you got to have, and you're also getting electrons in at the same time, and you're getting amino acids. So consuming stuff the way we do covers a lot of bases. But you didn't have to do it that way. It's possible to have life where you are consuming carbon in the form of carbon dioxide. You're consuming electrons in the form of different minerals and things, and you're using that system to actually operate and generate the molecules and the energy, particularly if you're not consuming things that have some energy in the bonds, the energy is coming from the sunlight. So we've got all organisms, and we're using chemical energy. You could use light. On the other hand, there are no pathogenic organisms that are using light, so we don't have to worry about those guys, happily. Let's just stick to this side. You got guys that the carbon source is carbon dioxide. These are the chemoautotrophs, and they are using a variety of different kinds of molecules as their final electron acceptor. But again, not pathogenic, so we don't have to worry about them. We got these guys. They actually are pretty similar to us. Chemoheterotrophs. The final electron acceptor can be oxygen, and that's us for sure. Most fungi and microorganisms like protozoa, amoeba, eukaryotes, pretty much all eukaryotes, and a bunch of bacteria, but not all. Most pathogens. You also then have guys that are not using oxygen as a final electron acceptor. And these are things like Streptococcus, some guys that may be pathogenic but not always. Using inorganic compounds, that's a fairly strange thing, but there are some guys, these are obligate anaerobes, and the organism that causes botulism is one of them. We pick on botulism a lot, Clostridium botulinum, because there are not a whole lot of obligate anaerobes. But it's one of the things we'll start seeing. The bacterium that causes tetanus, it only happens when you have a deep puncture wound, because you have to have an environment that doesn't have a lot of oxygen. And so if you get a deep puncture wound, you step on a nail or somebody stabs you, and then that wound seals up, you have introduced bacteria into that wound, and you have generated a somewhat anaerobic environment after a little while. And so you can end up with some fairly nasty bacteria growing in those kinds of closed wounds. That's why a puncture wound, a deep puncture wound, is something that to be taken a lot more seriously than a surface abrasion. All right. So, as I've mentioned, and I'm not sure everybody's here when I said this, I neglected to print out the lab and hand it to you, but recognizing the idea that really how many people are going to read the lab before lab? Hey. So we're going to be doing gram staining, and it is a protocol similar, but not quite the same, as a little bit of the staining we did last week, where you're going to take a loop full of bacteria, put it on a slide. We actually have to dry the slide down a little bit on the incinerator, and then we're going to use a series of stains. The way gram staining works is that gram-positive guys, that cell wall precludes the uptake of certain stains. And so gram-negatives will stain one color, and then the gram-positives will stain a slightly different color as we use a second stain. And so we have a decolorizing step and a rinsing thing. We got the protocol all set up for you. And so we'll look to see. Gram staining is actually a bit trickier than what we were doing last week. But hopefully we'll see some differences. And we'll see some, it's sort of a pink versus a purple. So it's not like, a huge red versus green. But with some skill, we can actually get a pretty good gram stain. And we'll see how that rolls. So I'll meet up with you guys at 1. Go have lunch. Go eat some fermentation products.